With the same class from where we left last class. We are continuing with the molecular biology short answers which you will get in the examination. So moving on to that, the first thing that we will discuss today is DNA markers. So what is the DNA markers? These are used for genetic mapping of the genome for the diagnostic purposes. There are four types of DNA sequences which can be used as markers. So these are restriction fragment length polymorphism, or we also call it as RFLPs. And then you have the mini satellites or variable number tandem repeats, or it is also called as VNTRS. And then you have the micro satellites or simple tandem repeats, that's STRs. And then you have the last one is a single nucleotide polymorphism, we also call it as SNPs. Okay, so these are the DNA markers. So moving on to each individual DNA marker. So among them, the first one is a restriction fragment length polymorphism or RFLPs. So this is in the human genome. We all know it is heterozygous. So these have a number of variations of DNA sequences. So these variations of DNA sequence, we call it as polymorphism. So in a normal healthy individual, these alterations occur only in the non-coding regions of the DNA. So any differences in the DNA sequence can result in variation of the restriction sites and therefore in the length of the restriction fragments. So an inherited difference in the pattern of restriction sites is known as a restriction fragment length polymorphism or RFLP. So this RFLP helps in the diagnosis of certain diseases. So when the RFLP lies within or even close to the locus of a gene that causes a particular disease, it is possible to trace the defective gene by analysis of the RFLP in the DNA. So it can help in reduction of a single gene based diseases with at least 95% certainty or positivity. Okay, so RFLP can be used to detect the human genetic diseases and in prospective parents or the fetal tissue if it is taken for diagnosing any genetic diseases. So example among them is sickle cell anemia, cystic fibrosis, retinoblastoma and Alzheimer's disease. And RFLPs can also be used in forensic medicine. So it is used to establish a unique pattern of DNA fragments by DNA fingerprinting of a particular individual. So it can be used in forensic medicine. So the most important application of RFLP is diagnosis of the disease and in forensic medicine, right? So we'll have a look at how the RFLP can be used. So the blood sample is taken, then the isolated WBCs are taken, and then the purity of the DNA is tested. Then the DNA is digested by the restricted endonuclease enzyme, and then the fragments are separated by electrophoresis, right? And then it is blotted by southern blotting technique and transfer of the DNA to this membrane, and it is labeled by DNA probe. And this DNA probe, it is labeled means then you do the auto radiograph and then the DNA is analyzed, and you can see there are different DNA fragments which are of different lengths. So this is the technique of RFLP. Next DNA marker you have is the variable number tandem repeats or VNTRS. So similar to RFLPs, VNTRs are DNA fragments of different length. The difference in them is that RFLP develops from random mutation at the site of restriction endonuclease activity, whereas VNTRs are formed due to different number of base sequences between 2 points of a DNA molecule. So VNTRs are composed of tandem repeats of short base sequence, and the number of elements in a region may vary. Hence, they are known as variable number tandem repeats. So the pattern of VNTRs and RFLP is used in DNA fingerprinting, as an individual genome has different VNTRs and RFLP which are unique to a particular individual. So VNTRs are also helpful in detecting the genetic diseases which are linked in changes in the degree of repetition of the microsatellites. Example, in Huntington's chorea is a disease observed when the VNTRs exceeds 40 repeat units, right? So that's one example. And when the DNA molecule are subjected to restriction endonuclease action, the VNTR sequences are released and they can be deducted due to the variability in the repeat sequence copies. So this is used in mapping of the genomes and DNA fingerprinting. Okay, so this is the second type of DNA marker. Then you have is a microsatellite, or we also call it as a simple tandem repeats, are of short repeats of units usually composed of dinucleotide or tetranucleotide units. So these simple tandem repeats are favored over the VNTRs as DNA markers because unlike VNTRs, they are evenly distributed throughout the genome. So further, when we subject it to PCR, can be used to identify the length of the polymorphism in that particular fragment of DNA, right? So this is the third type of DNA marker. Then the last one you have is a single nucleotide polymorphism, or we also call it as SNPs. These are located at the genome where some individuals have one nucleotide, that is for example the C, while the others will have a different nucleotide, example G. So there are large number of SNPs in the genome, and the human genome contains at least 3 millions of single nucleotide polymorphism, right? So there is a change in only the single nucleotide base. So in this slide you can see there is only a single nucleotide polymorphism. The microsatellite, you can see there is 5 repeats of the nucleotide bases, whereas only this particular nucleotide base is changed, that is T is changed to adenosine in both these DNA strands. So this is only a single nucleotide polymorphism. Okay, so DNA markers comes as a short note, so you have to write all the 4 types of DNA markers. Next, we'll go on to another short note that can be asked, that is DNA fingerprinting or DNA profiling. So what is it? When the microsatellite repeats are amplified and separated by the blotting technique from both the chromosomes, so 2 possible alleles are generated from each selected, then a pattern of these markers is generated. So when an individual's DNA is analyzed for such markers, it becomes unique to the individual to the level of a fingerprint. The set of such markers or alleles of an individual is called as a fingerprint, and the whole process of identifying the individual with the help of a microsatellite repeat allele typing, we call it as DNA fingerprinting. So what are the applications of this DNA fingerprinting? The DNA fingerprinting is used in forensic medicine to settle certain paternity dispute or identify an accused by matching the available sample of DNA from the sources such as blood stains, hair, skin, etc. And also it is used in immigration test cases and also certain disputes in this immigration, right? So this is the application of DNA fingerprinting. So in the next slide, we'll have a look at how this DNA fingerprinting process takes place. The blood sample is taken from the patient. Then the DNA is extracted. Then it is fragmented by using the restriction endonuclease enzyme. So you can see then after that the DNA fragment is subjected to electrophoresis and then it is subjected to blotting process, that is the southern blot, right? And then it is labeled with a radioactive DNA probe is prepared, and then it is hybridized with a DNA probe, right? And then it is washed, that is excess of the probe is taken off. Then the radioactivity bound to the DNA on the membrane can be seen, and then it is subjected to an X-ray film and it is placed next to the membrane. Then the film developed with the DNA fingerprinting pattern is obtained here, and it can be read and the identified. Okay, so this is the whole process of DNA fingerprinting. So DNA fingerprinting also comes as a short note. So please write in detail the theory part and also the flowchart, which is much easier to explain the whole process of DNA fingerprinting. Next, we'll go on to another short note that can be asked, that is RNAi or RNA interference. So what is RNA interference? It is an emerging as a new strategy to selectively shut off the post-transcriptional expression of mRNA. So the rapid, inexpensive and selective silencing of a gene product in a complex biological system, which is useful in fields of virology, cancer research, genetic disorders, drug designing, etc. It can be used. So what is the mechanism of the RNA interference? So the double-stranded RNA, that is dsRNA, when introduced or synthesized inside the mammalian cells, undergoes processing by an enzyme called as a dicer. Now after this, it will result in a formation of a 21 nucleotide base pairs of a length that is called as a small interfering RNAs, or we call it as siRNAs. Okay, this siRNA gets coupled with a cellular protein to form the RNA interfering silencing complex, that is RSIC, and this recognizes the target on the intracellular mRNA and effectively leads to cleavage of the mRNA, right? So what happens after this? The short synthetic double-stranded siRNA can be easily introduced into the mammalian cells using transfecting agents. So it can also be expressed intracellularly when introduced through the plasmid or the viral vectors. So siRNA should be around 21 nucleotide base pair length long with 3' overhanging of 2 nucleotides rich, that is the GC content should be more, and the complementary to 50 to 100 nucleotide downstream of initiation codon of the target mRNA. And this siRNA are introduced into the cells by using plasmids or the viral vectors. So what are the clinical aspects of RNAi? So it represents new potential targets for therapeutic drug development in humans. So it is also used to kill cancer cells in the mice by using mini cells, EDVs, we call it as gene IC delivery vehicle. So the first wave of mini cells release the siRNAs, right? So these, which switches off the production of the proteins that makes the cancer cells resistant to chemotherapy. Then a second wave of EDV cells, then accepted by the cancer cells, releases the chemotherapy drug by killing the cancer cell. Okay, so in the next slide we can have a look at how the RNAi helps in cleavage of the mRNA. So the double-stranded RNA, you can see here dsRNA, with the help of a dicer, it leads to the formation of small interfering RNA, which is made up of 21 nucleotide base pairs. So this combines with a cellular protein to form the RNA interfering silencing complex, and this recognizes the target recognition and it acts on the target mRNA and cleaves the mRNA into two strands. Okay, so this is the application and the clinical aspect of how the RNA interference is used in the newer science technologies to help in certain diseases, right? Then the next, another short note which can be asked in the exam is um, AQUASOMES. Okay, so now what is AQUASOMES? These are nanoparticle carrier systems with three layered self-assembled structures. They comprise the central solid nanocrystalline core coated with polyhydroxy oligomers onto which biochemically active molecules are absorbed. So the solid core provides a structural stability. The carbohydrate coating stabilizes the biochemically active molecules. So because of this property of maintaining the conformational integrity of the bioactive molecule, the aquasomes are being processed as a carrier system for delivery of the peptide-based pharmaceuticals. So the delivery system has been successfully utilized for the delivery of insulin, hemoglobin and various antigens, right? So the oral delivery of peptidase has also been achieved by these aquasomes. Okay, so this is about the few short notes which we have discussed in some of the topics, but not to an extent how to write a short note, right? So this is about the molecular biology chapter as a whole. So we've already done with one set of assignments in molecular biology. So you have another set of questions that comes up for today's class. So that please write it and please send it to us. So this assignment, you have long note and short essays. So one long note that can come here is define recombinant DNA technology, discuss in details and add a note on applications. Okay, then the short essays, define hybrid, chimeric or recombinant DNA, and then you have the list and explain the role of vectors in recombinant DNA technology. Explain the formation of cDNA, define and discuss the process of PCR. Then you have the state the importance of northern blot technique, explain southern blot technique and its applications, state the importance of western blot and its applications. So most of these processes, please see to that you write the theory and also draw the diagram, right? Then you have explain briefly DNA microarray technique and its applications, enumerate the DNA markers, SNP, VNTRs, RFLP and state its importance. What is RFLP? Mention its clinical importance. What is gene therapy? Name vectors used for gene therapy and give examples. Explain the basis of DNA fingerprinting with the help of a neat labeled diagram. Discuss the therapeutic role of RNAi. Then give an overview of Human Genome Project. Define and illustrate the role of DNA probes. What is tumor markers? So you have classified tumor markers, giving examples. So this assignment will have both the radioisotope and also the cancer chapter. Okay. Then the next question is radioisotopes and their applications in medicine. Define cancer and enumerate the causes of cancer, including physical, chemical, genetic, and biological. Elaborate the role of mutagens and anti-mutagens in the etiology of cancer. Explain the action of oncogenes and anti-oncogenes. Describe the role of oncogenic viruses and association with cancer. Enumerate diagnostic and prognostic applications of tumor markers, including their elevation in benign and malignancy conditions. Okay, so this is another set of assignment along with molecular biology, cancer and radioisotopes. Okay, so this finishes for today the molecular biology topic, along with it cancer and radioisotopes, right? So thank you. Have a nice day.